Today's topic is vision. Now, after knowing the layers and the styles of retina, see, there's a chemical process, that is a process how which you perceive the vision. There is a lot that happens, that is, for example, for initiation of a visual impulse, there what happens is there's a photoisomerization and hydrolysis of the visual rods and cones, and this is the process, not much important. But what do you, that is a rhodopsin will get converted. Rhodopsin, which is a combination of opsin and retinal, or 11 cis retinaldehyde, is converted to 11 cis retinol, and after the G protein coupled receptor is activated, the cGMP. Now usually in a cell, when there's a stimulus, there is depolarization. So when you open the sodium channel, everything moves out, so it is depolarized. But this doesn't happen in visual impulse. What happens when the light strikes, the sodium channel will close. This happens in the during the day, sodium channel will close, so hyperpolarization will happen. If this hyperpolarization happens, then the stimulus is received, and then further it is action potentials are generated, and this happens by reducing the cGMP. So decreased neurotransmitter release, and the bipolar cells will respond. But in case of dark, what happens is the sodium channels will open. The sodium channels will open, the cGMP will rise, and then the neurotransmitters will be increased. There is depolarization happens, and another type of response. This is what happens here. And now between the rods and cones, the rods, which are also known as visual purple, they are mainly photosensitive receptors. They're mainly photosensitive receptors, 120 million in number, and cones, used for color vision, acuity, etc. Now these rods and cones will release glutamate. Now the rods and cones are the primary, there is initial primary neuron. Now if you see here, in the retina, the optic disc, this is the FS means foveola, M is macula. In optic disc, there is no photoreceptors, there is no rods, and there is no cones. In fovea, there's only cones, and in the macula, you have a decent amount of receptors. But in the peripheral, the rods will be more, and in fovea, the cones will be more. So this is what this is regarding the location of the photoreceptors. Now this happens, what happens is, from the impulses from rods and cones will go back to the polar, which is the second order neuron. First order neurons, rods and cones. Second order neurons is bipolar. Third order neuron will go to the optic nerve, that is the ganglion cell layer. Fourth order neuron will go to the lateral geniculate body, or optic radiation. Now all these will show local in all the types of cells that are present here, local potentials only are generated, but only in the ganglion cell layer action potential will be generated. So the only cell where ganglion is action potential, and similarly the only cell which we told that everywhere hyperpolarization happens, the only cell where depolarization happens is the amacrine cells. These amacrine cells will also secrete ACH, indolamine, dopamine, GABA, glycine, everything it will secrete. Now what happens after it reaches the lateral geniculate body? Now lateral geniculate body, now it has to go to the thalamus. In the thalamus, not thalamus. After going to the lateral geniculate body, it goes to the thalamus. And the thalamus, there are 6 layers. So 6 layers, 1, 2, 3, 4, 5, 6. The 1 and 2 are known as magnocellular pathway, which is responsible for sensing the eyeball movement, or everything eyeball movement related senses are relayed there in the thalamus. And in the 3, 4, 5, 6, the parvocellular pathway, the color vision will go and relay there. Now everything, see, if this is the right eye and this is the left eye, from the contralateral eye, all these senses are going to go and relay on 1, 4, and 6. In the ipsilateral, it goes to 2, 3, and 5. And this is what happens. Now most of the lateral geniculate body senses or neurons will all terminate in the layer 4, especially layer 4C. So this is regarding the magnocellular and the parvocellular pathway. Yeah, that's it. Also, yeah, one more fact is that, see, funny currents we've already read in heart, which is present in the SA node, uh, which produces the I think the phase 3 action potential of the atria. In eye also it produce the funny currents are present, which is present in the retina, especially in the rods. So this is also present here. Yeah, and about the one word about all the cells, the rods are for vision, dim vision, cones are for bright light and color vision, bipolar cells, not much to say, ganglion cells, which forms the optic neuron, and it is only action potential producing cell, it is only output cell. See, everything from all the input is converted to output in the ganglion cell. And other few other cells like horizontal cell, amacrine cells are also there. And there's no role in vision. There's a cell which has no role in vision. It's known as retinal glial cells. They only support the retina, which is also known as Muller cell. They don't have any role in vision. They just support the retina. So all these are regarding vision.